Welcome to this session on next-gen AAV vectors for gene therapy. I'm Gula Lifshitz, a senior biotech analyst at Chardon. So therapeutic viral vectors using natural and other extensively studied capsids such as AAV9, AAV5, and others have shown pretty transformative effects in the clinic and are continuing to emerge in the commercial arena with a few approvals this year. However, the field has also learned some limitations of these vectors relating to things like biodistribution, the type of cells they can transduce, immune response, manufacturability, and other parameters that we've all been discussing here. A greater understanding of these obstacles and how AAV biology interacts with human biology, as well as advances in enabling technologies such as high-throughput sequencing and machine learning are enabling progress on this front. So we'll be discussing these issues with our participants who are developing next-gen platforms. I'm joined by, in a random order seemingly, Rick Modi, CEO of Affinia Therapeutics, Unique Mehra, CBO and CFO of 4D Molecular Therapeutics, Peter Anastasiou, CEO of Capsids Therapeutics, and Adrian Veras, CSO of Dino Therapeutics. Welcome to all of our participants. So I said some comments in the beginning about the trials and tribulations of AAV, but I think framing the problem would be better coming from you. I'd like each of our participants to briefly introduce themselves and frame the problem. What do you see as the major challenges that you're trying to address, and what is your approach? And maybe we can start here and work our way down.
Yeah. Well, thanks for the opportunity to be here. I'm Peter Anastasiou, the CEO of Capsids. From our perspective, the main challenge we're trying to address is delivery, and to deliver, especially in CNS disorders or other biological other organs with biological barriers for that matter, to be able to transduce those barriers and to be able to affect cells like in the brain, neurons in particular, at high enough quantities that you can impact diseases that are more common and across age ranges, and at the same time being able to do that while detargeting the liver to avoid some of the safety issues that have existed with first generation technologies, and then finally do all that with IV administration.
I'm Rick Modi, the CEO for Affinia Therapeutics. I think you're going to hear quite a consistency here. So we founded the company about 3 years ago with the reckoning that many of these therapeutic payloads were going to have one particular issue, and that was one of delivery. Thank you, Peter. The way we're approaching it is to take conventional AAVs, recognize their limitations on 3 or 4 different attributes. That includes tropism, manufacturability, immune phenotype, and translatability, and then work on those 4 attributes in a very methodical, rationally designed manner. The 2 areas that we're working on is CNS. Again, similar to what Peter described, it's one of the areas in which we think we can unlock many large diseases in adult populations, and also in muscle with a very similar approach, large diseases, and be able to unlock many different diseases. So, pleasure to be here. Thank you for inviting us.
Thanks. I'm Unique Mehra with 4D Molecular Therapeutics. Again, continuing on the same theme, one of the biggest challenges with conventional, naturally occurring vectors is delivery. So our approach is basically using directed evolution platform or technology to create customized vectors that are modular, proprietary, can be customized for any disease or therapeutic area that we intend to pursue. And the main attributes we're looking to resolve is delivery transduction as well as quality of gene expression into the target tissues, basically. Thank you.
Hi everyone, so I'm Adrian Veras, I'm the CSO of Dino Therapeutics. So our Dino is about 5 years old. We're slightly more than 100 people, and we're 100% focused on capsid engineering. Our approach is to combine proprietary NHP in vivo data with AI models to analyze this data and design new capsids that iteratively improve across all the properties which are essential for solving gene delivery. Our focus is on creating the capsids that are needed by you all. And what I mean by this is we're not developing our own therapeutics internally. So we're 100% focused on a partner-centric model, where our approach is to license our capsids to enable our partners to work at the leading edge of gene delivery. And so it's a unique business model where we're trying to ensure that we succeed only when our partners succeed, and we're here to do that.
Great. So sounds like delivery is important. We got that. in terms of where the space is now, capsid engineering has been around as a concept for over 15 at least years. So I'd like to understand a little bit more about how the discovery platforms are different now from early capsid engineering effort. Like, for example, in the past few years at the ASGC conference this year, there was a bunch of companies and academic groups describing various screening workflows using a library of vectors that are diversified from some parental capsid, then transduced into some system, and they look for transduction of their favorite cell type or crossing the blood-brain barrier. So for those of us looking at this space from the outside, number one, how is this space different? And number two, how do we distinguish these strategies and understand how the different choices of the parameters that are being used can impact the type of vectors that you get out? And maybe we can start with Adrian and answer from there.
Yeah, so there's a lot of activity in the field. As we said, delivery is a very important challenge. The key aspect, the two inflection points in the past 10 years have really been the advent of multiplexing readouts that enable us to test hundreds of thousands of capsid at a time, as well as computational models, which allow us to predict capsid properties and efficiently explore all the myriad of possible sequences for AAVs. So one of the key ways in which platforms differ is where mutations are made and how. So for instance, most library experiments are created using randomness. In our approach, we use AI models to design every sequence that we test and thus have a very high success rate in terms of measuring it in vivo. I'd say that as somebody who's thinking about using capsids, you don't have to compare platforms. You should actually focus on comparing capsids. And what are the properties on a per capsid basis of what's the percent of cells that's achieved in a particular tissue of interest and at what dose. And that's ultimately what's going to matter from a clinical efficacy and safety standpoint.
Go ahead, anyone jump in.
Yeah, we believe at 4DMT that I think you need diversity in your initial set of vectors to really choose from. So we use directed evolution to really distill down from 1 billion synthetic capsids, which have all been molecularly adjusted from the naturally occurring capsids. And then we use a process of competitive selection. So we define what is the target vector profile we need to achieve, what is it that we're really going, including what is the mode of administration to the desired tissue. So we'll lock in all of those attributes, and then we competitively sort of select through a process of iterations. We've consciously done that in NHPs, in non-human primates, because we believe that's the closest translational model to human beings. And we've got great success in doing that because 3 of our vectors are now in the clinic and validated. So ultimately, when you look at that, now of course you have more, as Adrian mentioned, computational power coming into these screening platforms to distill from that diversity, as well as now you more and better understanding of molecular constructs to be able to sort of bring about the variety and from which to distill on.
Any other comments from you?
Yeah, I think maybe I'll take a bit of an answer there. I think there were 2 questions there. One is, how are you all different? And then the second, why would I believe you that you can produce something better than AAV9, which has been around 15 years? I do believe there's many different ways of finding new capsids. I don't think there is a right or a wrong. I think if you just listen to all of us, we all have some different approach behind it. But I do believe all of us are leveraging the best cutting-edge technologies that are out there: next-gen sequencing, single-cell analytics, machine learning, for example, receptor identification as well. Those are the things that were not around 15 years ago, or 10 years ago, or even 5 years ago. And if you compare different capsids from different companies, you'll see the improvement in tropism that we're now getting from these capsids is not an incremental change, but a transformative change. Where AAV9 was getting us 1 or 2% of neurons in adult monkeys, for example, we're now north of 50%. Same thing in muscle. If you look at what AAV9 was doing when given IV, it was high single-digit % of myocytes. We're now north of 50%. That is the proof that what we're doing will actually get us beyond the small diseases that are out there. Beyond the platform comparisons, there are also differences in company strategy as well. Again, some companies opt to focus just on CNS. We're doing muscle and CNS both. We intentionally forewent, for example, liver and ophthalmology as an example. So there are also differences in just company strategy. Though. Again, not to say that it's right or wrong, but these are different stances that companies take.
Yeah, and I agree, there's a lot of similarities. There's also differences between what we all do, but there's certainly enough unmet need out there, and we're all taking a slightly different approach, and even for those of us who are taking similar approaches are going after different therapeutic areas and different targets. So I certainly wish all these companies and every other company in this space good luck, because we all benefit from it, and ultimately patients benefit from it. So that's the most important part. To talk a little bit about in the past, so our technology came from Caltech. And at Caltech in Viviana Gradinaru's lab, the engineering work was done in mice. And so when it came over, when we started the company, we transferred into an NHP-driven, but we still do preclinical mouse models, so we kind of back translate to the mouse, but also we do human clone cells to improve the greatest chance of having translatability. And what we do is we start with the disease in mind. One capsid does not fit all, and every disease requires something different. So what is the organ you're trying to impact? Within our main target organ of interest, the brain, which for this disease, what part of the brain do you want to impact? What types of cells within those parts of the brain are you trying to impact? And of course, all the off-target effects. One of the main foundational patents that we have is detargeting the liver, and then we filed another 40 or so patents since then. So a big part of our screening is not just getting that organ and cell type specificity, but then also getting all the off-target effects out. And one of the other things that's enabling for us has been the high throughput screening from automation and robotics that we do. So we literally start using directed evolution with billions of capsids and filter down to that one single capsid that's focused for each disease, and we're able to do that by sifting through large volumes, hundreds of thousands of tissue samples to make sure that we're getting to the right profile that we're looking for, and the robotics and the automation is what enables that.
Great. So you touched on a couple of points there, both in terms of the type of indications that could be targeted, as well as the granularity of different cell types within a given organ or tissue that can be addressed. To what extent, are these issues the next frontiers in this space? Where do you think, where do you think the field can go in terms of granularity in the different cell types, and what does that mean for therapeutic applications? Unique, you can say.
Yeah. So I think, look, getting into very specific cell type tropism, I think it's challenging still for the industry right now. I mean, you can do certain sort of framework around, introduce regulatory promoters. You can do very tissue-specific promoters that you can work on, and I'm sure a lot of companies are doing that right now. You can also, the more targeted your therapy is, the better the tropism is. I think some of those activities, Julia, are underway, and we still have a long ways to go. But I think the industry, based on all the work collectively that's happening and the understanding of not just molecular structures but genetics, is facilitating a move towards that. I think we still have to sort of take a few more steps to get to specific tropism in each of the tissues.
I also think we're making a lot of progress, and we're really on the cusp of, and really at some major breakthroughs, not just our company, but other companies as well. And as an example, some of the data that we presented at the ASGCT shows that we also are getting well north of 50% in many brain regions, and as high as almost 70% transduction of purely neurons. So having that cell type specificity where we're not also then having impact simultaneously on glial cells or astrocytes or other things, because at least for the target disease that we did that capsid development for requires that neuronal specificity. And so I think we're there now, and at the same time having liver detargeting. And so that same data that I'm referring to basically showed that from DNA expression or RNA expression, you get anywhere from 500 to 1,000-fold greater CNS to liver tropism versus AAV9. And so that kind of transduction, that kind of specificity really enables a lot of diseases that are broader in prevalence, but also broader in terms of age ranges as well.
So Rick, you mentioned receptor identification. How does that play into these issues of the type of cells that can be transduced, the type of indication that we can go after?
Yeah, it's a good question. I think in the two examples that we have been working in, if I take the muscle examples, much of the work which we did for Vertex as an example, specific for Duchenne muscular dystrophy and myotonic dystrophy. In that case, we had structure-activity relationship in hand. We developed the libraries. We put into NHPs, demonstrated the data that we did for myocytes, and similarly, liver detargeting and DRG detargeting at a considerably lower dose. On the back end, we identified the receptors. Now, the identification of the receptor in muscle is important, just as in CNS and many other tissues, for a few reasons. One, it actually de-risks significantly your human risk. In the case of muscle, we identified it was the integrin class of receptors, particularly alpha V beta 6 as an example. We know that integrin is conserved across mouse, monkey, and human beings. And we've done additional studies in human myocytes to demonstrate that this risk going to the clinic is significantly less. In CNS, it is a bit similar, although in some cases differently. Here, again, given the progress we had made, we have a short list of 15 receptors in mind that may enable the transcytosis of AAV across the blood-brain barrier. This is very powerful. So we're starting with synthetic libraries based on structural modeling in exactly how these AAV can dock onto the receptors and bind to it, and then moving into in vitro and in vivo studies. This actually changes the entire paradigm of AAV discovery into classical drug discovery that small molecule folks have been doing. It's actually a game changer for years to come. And again, similarly, it de-risks things in human studies coming in when we go into the clinic where these receptors are conserved in human beings, which is how we start by identifying the short list of 15.
Maybe Adrian, to what extent can AI and these machine learning approaches help move the space in this direction in terms of moving things from an empirical screening standpoint to more of a programmable modality?
Yeah, so AI is central to everything we do. From the get-go, when we started Dino, we've had some of the foundational papers in the field using generative AI to design capsids, going as far back as 2017 when we were still an academic project at Harvard. The key concept here is this first law of screening, that you're going to get what you screened for. And one of the challenges with direct biological screens is that you can only really select one modality at a time. Now, computational models and AI allow you to actually look at several properties at the same time and optimize for them in parallel. It's actually why we target libraries that are at a size where we can measure every property for every variant in this library. That's what gives us the training data that's most important to create new capsids. The final thing that AI really gives you is the ability to create truly novel capsids. So looking at editing the deeper core of the capsid, making as many changes as possible there, really the space of capsids is enormously vast. And ultimately, many of those changes are going to be necessary for us to break through things like neutralizing immunity and reach as many patients as possible. So it's really a foundational technology in the field.
To think about safety now, you talked a little bit about translatability and moving into non-human primate models as those are closer to human, et cetera. Where are we in terms of predicting issues regarding safety in various preclinical models, whether it's non-human primates or some human cell systems, for example, looking at things like complement activation or some of the other safety signals that have emerged in clinical studies of AAV so far? You can start here.
Sure. I think that there's a couple things from our perspective that address that, and most of it is specificity. So I know we're repeating some of these points, but specificity also is very relevant, not just to the efficacy discussion, but to the safety discussion. So like in our case, being very neuronal focused versus, let's say, involving glial cells reduces the immune response. Also making sure that we have that focused transduction versus of the target organ versus off-target organs, like the quote that I gave you about the 1,000-fold difference between CNS and liver detargeting or liver targeting relative to AAV9. And then the same is true for other organs like the spleen or DRGs and others. And so the more you can be specific, both in terms of cell type and an organ type, I do think it addresses and prevents some of the immune responses that can be seen with something that's more widely, I guess targeted is not the right word, but widely distributed, despite being IV administered.
I think, Gula, I would say, if you really think about the things that we have seen with AAV, it's actually incredibly well characterized by now. And many of these things you can now predict. Some you can't. We'll get to complement activation as one example of that potentially. But hepatotoxicity has been known. So why do you hear all of us being able to transfuse the liver 10,000 less? Because we know that is an issue. You actually see hepatic transduction in monkeys well before you put the product into human beings. We can actually also see transaminases go up in NHPs. So in the detargeting that we are doing, we're able to see that we do not see transaminase elevations when we transduce the liver significantly less. Again, DRG, inflammation, or toxicity, however you want to describe it. You can use nerve conduction, including in NHPs, to be able to look for that. And we haven't seen much of that in humans as an example. Complement activation is a bit tricky, to be honest. It's not that there is a predictive assay, unfortunately, yet developed. We do have one where we have positive controls, which if you use enough samples, you do see a positive signal. And again, we put our capsids in that type of an assay and gotten comforting data that we would not see that in the clinic potentially. But that requires, because that event rate is so low until maybe some recent capsids, in that case, you need many large number of sample sizes to look at for a positive control, right? Again, these are quite well-characterized AE events that we are either designing around structurally on the capsid or being able to screen for in monkeys and then be able to look for in human beings in the clinic via good surveillance. In some of these cases, you can preempt them with pharmacologic treatment or treat them subsequent to it, and which is why indication selection is so important, right? So if you're going after a deadly, rapidly progressing disease, then the benefit risk for a therapy like this is warranted. If you're going towards a more disabling disease, perhaps one might want to consider about indication choice, right? But I think it's a mix of all of the above things.
Yeah, I think the only other point to add to what sort of Peter and Rick have said is also building on the specificity, you also have to consider novel immunosuppressive regimens, right? I mean, because there's a part of the characterization that we all know, but there are the untold effects as you translate from the monkeys to the humans. So I think you have to also say, what is the lowest dose? What is the immunosuppressive regimen that can help you get through that, as well as avoid the sort of immune reaction? I think that's another consideration that's pretty important in the equation.
So what I want to echo is this point of low dose. So I think one of the key things that this next generation of AAVs enable is transducing many cells at a low dose and actually tailoring that appropriately for a given payload, for a given indication. So for example, our Dino-BCAP1, a CNS-directed capsid that crosses the blood-brain barrier, produces 5 to 20% of neurons and cynos at a 1E13 dose. So it's creating you the optionality of you can go up by increasing the dose, or depending on the payload in certain disease indications, you can stay at that lower targeting. Another key aspect here from safety is this notion of translatability. So we really favor looking across NHP species. I think cynos are the core model to do screening in, and then also confirming that capsids work, for example, in AGMs, really helps us translate through the challenges of from non-human primates to humans.
And to what extent do you think evasion of immune responses that have been part of the kind of the screening elements in a lot of programs, to what extent do we think that's feasibly going to enable redosing of AAVs, or is that something that's still a little bit further away in terms of understanding and solving? Rick, you sound like you have a thought.
I can start, and I'm sure my colleagues will build on it. I think solvable is an English word, so I think it's solvable, the problem of PEI. If it's not an English word, we'll create one today, at least for this panel. There are a few ways to get there. When we screen capsids and we design them, we of course have a multiplex neutralizing antibody assay that we do, and our capsids are at least as good as AAV9, if not better. We haven't done optimizing for screening against pre-existing immunity. We can optimize it further. But there are a few ways actually to solve around this. One of them is if you do have a high PEI, either because of a prior dosing or just innate PEI, you can wash out the antibodies. People have looked at plasmapheresis. There are new novel enzymatic interventions that can be employed also to clear out the PEIs as an example. You can also block it. You can block it either prior to the first exposure of the vector with a triple immunosuppressant regimen, which is used quite frequently in many other diseases as well, or you can block it on a secondary opportunity as well when you're potentially redosing. And then the last one, not necessarily in this order, is potentially you could also design around it from a structure perspective, both in the capsid where you can put in significant sequence diversity and aim to go for a lower cross reactivity versus the conventional serotypes, or potentially in the transgene where you can mute the initial immune response as well. And I think between all these things, I do believe it is solvable.
Just to follow up, I certainly agree with the point that there's emerging technologies that can help with the redosing. But I will hit the point that's kind of implied in your question, and that is the presence of pre-existing antibodies in animals, but also patients. And one of the things, because we do a big part of our screening is the immunogenicity platform of these billions, or a profile of these billions of variants, we have seen with some of the data that we presented at ASGCT that there's not a marked difference in terms of transduction in animals that had pre-existing antibodies versus those that don't. So that gives me hope that, and belief that at least on the near-term part of the equation, which is, how can you administer these therapies to patients that have pre-existing antibodies, is something that can be addressed with some of the screening that we've done. The other thing I would add is that there could be screening in other serotype, or engineering in other serotypes that maybe have less pre-existing antibodies for patients, like an AAV5, that would enable that part of the equation. But yes, the redosing certainly hasn't been solved, but I think there's a lot of technologies that can help us solve it.
Any additional comments?
The thing I'd like is this modeling, the thinking about this problem really and what the capsid can do and what the non-capsid can do, and really bringing to bear the combination of those components. It's clear that the capsid plays a direct role. The reason we know this is the variation amongst natural serotypes of pre-existing immunity, and being able to dial that down across engineered variants. So immune evasion is one of the key properties of every capsid in our libraries that we measure, and we integrate this into our designs. It's also a reason for us, our platform isn't focused on just AAV9, but actually on the full breadth of natural serotypes that we design using our models, because ultimately there's some combination there amongst those the learnings from those different serotypes, which are going to be essential for this question, which is longer term for the field. Delivery is first, but this evasion question is really coming up right after this generation of capsid solves delivery.
Anything to add?
Yeah, the only thing is, I think it comes back to designing around that right from the get-go. We've been able to see that we get asked this question quite often in our lung sort of a portfolio, to say, redosing is an important consideration. The vector that we characterized was designed to account for that. In fact, in our first cohort of 3 patients, 2 of them had antibodies neutralizing, and we were able to still get the 92 to 99% expression. So that shows that there's promise in designing around that. And then I think to build on everybody else's point, that then the second aspect kicks in: how do you control the gene expression to the specific tissue there? I think they all build into that point.
Got it. So maybe just to back up a bit, there's clearly a lot of activity in the space, a lot of diversity in terms of the strategies, and a lot of diversity in terms of the vectors that are being generated. So how many different AAVs do we need? Are we moving towards a model where each franchise will have its own proprietary AAV? Do we lose some of the platformness of the technology as the field kind of expands? How should we be thinking about the diversity that is optimal within the AAV space?
So people were probably asking that same question 30 years ago about antibodies and about biologics. Is the first company that solves this gonna basically, solve everything? And I think we now know how that story ended, which is that one size doesn't fit all. And I think the same is true here. And at least from our perspective, we clearly don't believe one size fits all. Even within our own platform, every single disease that we're targeting either with our own wholly owned programs or with a partner has a bespoke profile that's created of what's needed and what we're looking for. So we don't even have that one size fits all approach within our own company, let alone among all the other companies. And everybody's learnings will continue to build on each other, and then we'll get more and more therapies from just a handful of the indications represented by these companies to dozens and dozens and hundreds of indications. So anyway, that's my perspective anyway.
There's so many patients and diseases that are in need out there. I think you're seeing 4 parties over here. That could be double the number of parties over here, and we still would have an unmet need out there. So I am comforted and believe that we could get more capsids. In our company, we do take an approach where we do look for diminishing returns, and we move on once we believe we have a best-in-class capsid. In muscle, we are there, we believe, for example, meaning any additional squeeze is not worth the juice to it, just to be very open. In muscle, we think it's probably 1 or 2 capsids to cover the skeletal muscle and the cardiac muscle, right, as an example. In CNS, it's probably 2 or 3 different profiles to be able to get deep brain versus cortical and the different cell types that could unlock many of these diseases. The reason we develop it like that is because we also believe that once you have a capsid in hand, you can make a platform out of that capsid. So take that one particular capsid, optimize it into the gene cassette, and then take it into a myriad of cardiac diseases or skeletal muscle diseases or CNS diseases. And there's great learning and efficiency in that. So again, it's not right or wrong, but that's the way we've chosen to look at the landscape. There are many, we talked to multiple partners out there. And one of the things they look for, of course, is that the field is available. So outside of the internal pipeline, also there are multiple additional players out there looking to make a difference to these patient communities multiple times over in the same disease too, right?
The only point to add further is, I think the question should be slightly different. It's not the number of capsids that we should be concerned about right now, it's the quality of capsids, right? I mean, we are still in that early phase in the gene therapy inflection. I think Peter mentioned, you look at the trend with small molecules, monoclonal antibodies, we are still probably in the first couple of innings of the gene therapy. So I think that question will probably be relevant a little bit down the line when the gene therapy is established itself.
I think my views on this, we're in the early phase as well. The way I think about it is, I think as long as you can't deliver to every patient, practically to every cell at the lowest possible dose, there's room for additional capsids. And we're also going to see that different indications will have different preferences amongst capsids, and there's going to be room for a substantial number of them. In every case, what matters most is going to be safety and efficacy. I also think we're quite a ways away from real diminishing returns and I think the real cap there is actually, if you think about the number of cells we're hitting on a per capsid basis when we're injecting, even at our low doses of 1E13, there's still many sort of log orders to go in terms of capsid improvements, which would all translate down to lower cost, which would be very meaningful for the field, as well as better safety. And we're there to go and create that benefit for gene therapy developers.
If I may add one more point,
Please
the only differing view I have from my colleagues is that we're emphasizing a lot the capsid, but capsid is only one component of the drug product. The other component is the cargo. And every capsid cargo creates a different drug product. And so part of the reason that we have that kind of specificity and don't believe one size fits all, not just for the disease and the target and what you're trying to get to and what kind of cells you're trying to transduce, it's also what cargo are you carrying. So the same capsid with different cargoes are 2 different drug products. And I think we have to think of it that way. I think regulatory agencies will think of it that way, certainly. And one of the things that I'm proud of is that for our wholly owned programs, we're focused on gene replacement, but with our partners, we get like our partnership with CRISPR Therapeutics, we have gene editing cargoes. Our 2 partners, Lilly and our 2 other partners, Lilly and AbbVie, don't want us to disclose the cargo, but these are yet other cargoes. So there's at least 3 or 4 different cargoes that we've had experience with pairing our capsids that I think kind of forms our view that one size does not fit all.
So this gets to a point that I was just about to touch on, which is the partnerships. And a number of you talked about it already a little bit, but clearly this is a space that pharma is interested in. We're also seeing partnerships with other biotech companies with complementary technologies. So can you expand a little on what you're seeing in terms of what partnerships are looking like, what partners are looking for in pairing with AAV players like yourselves? Can start with Adrian.
So at Dino, we have, so partnerships are central to our core business model. We have partnerships now with Novartis, with Roche, with Astellas, as well as with Sarepta. And we're very excited about these partnerships because they're how we translate our work into impact in the field. I'd say what partners are looking for, whether they're large pharma or small biotech, tends to be the same thing, which is they're looking to work at the leading edge of gene delivery. And that's what we are there to offer access to. We can work with new partners in any new partner in any new indication of choice. And what's helpful to flag there is also what we look for in a partner is actually commitment to the field of gene therapy, which is easier now than previously. But that commitment translating to a desire to really move aggressively with the vectors that are created and look to problem solve about what is new at this gene delivery frontier, whether it's regulatory, manufacturing, and working together to advance these products given the unmet patient need that's been described already.
Yeah, I think it's similar to what Adrian said, but also building on what Peter said earlier in terms of, I think the whole gene therapy is viewed as a modularity. So you have one side the vector, the capsid, you have the cargo on the other end. So I think the partnerships are coming in, especially interest to us in terms of the delivery, which is the novel capsid. But we internally are looking for where there are ways to enhance the cargo, whether it's gene editing capabilities on the cargo, which is probably one of the next frontiers in terms of how you express the gene. So I think it's a mix of those. Again, to Adrian's point, big pharma is always looking, having worked there for 13 years as part of Novartis, you're always looking at the next leading edge of, in this case, delivery. And from our perspective, we're looking at how we can sort of complement the delivery with other sort of modalities in terms of either the cargo or the overall capabilities that a partner brings.
I can go next. When we did the deal with Vertex, it was the time when we're still forming the company. We were in dialogues with like 5 other companies at the same time. So why did we marry each other, if I might call it that? They were eager to do something in particularly the 2 muscle diseases that we were choosing to work in initially. They had the best-in-class capabilities there with the deal they had done already with the acquisition they had with Exonics. They were committed to AAV in those 2 diseases. And they looked at the landscape and loved the work that Luc Vandenberg had done, which is what we in-licensed in the company. So that was the matchup. We were looking for a large upfront and a very low footprint on the company, and that's exactly what we got with them, just to be very open. There's a saying in the field, the worst thing than doing a BD deal is doing a bad BD deal. So we opted to put the number of diseases we have given up small and take the majority of the upfront that we could. But we worked amazingly close together. The people that are on the other side are phenomenal in AAV gene therapy. John Gray developed, for example, triple transfection way back when, as one example. So that's been excellent for us. Now when we speak with companies, they look at the CNS footprint that we have given up, which is 0, and they like the fact that we have these capsids and we haven't licensed those fields out, right? Again, we'll take a measured approach with it and make hopefully the right decision for what we want to keep internally for our pipeline and what we want to give up. Dilution, whether it's equity or pipeline, is still dilution. So one has to think like that, I believe.
I think there's been a tremendous amount of interest. When we ran our partnering process, we had more term sheets than we ended up acting upon. So that speaks to the interest. And I think the interest comes from the understanding from pharma that all the issues that have plagued gene therapy with first generation and why many investors, it's fallen out of favor, is because of the limitations of naturally occurring viruses. And so they understand that engineered viruses and the various approaches we all offer is the way to actualize the potential of gene therapy. And so we've had a tremendous amount of interest and consummated the deals with AbbVie and Lilly in CNS, and our partnership went so well with AbbVie that we expanded it into a new therapeutic area at the beginning of the year in ophthalmology. So while we're focused on, primarily the CNS, our engineering it can be directed at crossing any biological barrier, and most organs have a biological barrier, none as daunting as the blood-brain barrier. And so we're now starting to do that kind of work in ophthalmology. So I think they know that that's the way to get across the finish line in a lot of these diseases.
So in the last few minutes, I wanted to take a forward-looking approach. I'd like to ask each of you what you think the field will look like 5 years from now. Which challenges that we're facing now might be addressed? What are the current open questions that might still be open? Anything in that area, and we can start with whoever wants to speak first, or I'll call on someone. All right, Adrian.
I think there's this saying that your best shot at predicting the future is to try to create it. And I think what the future looks like in about 5 years is a lot of activity in more advanced clinical programs using the generation of capsids that exist and are available really today. In addition, the leading edge of what's possible with those capsids in terms of dose, in terms of percent cells, in terms of neutralizing immunity evasion will have progressed. And there will be a question of how are those brought, what's the path to bring those into the clinic? I also think what's going to change pretty dramatically in the next 5 years is also what other aspects of a gene therapy are going to be designed and how. For now, we're really still repurposing most components of a gene therapy coming directly from sequences naturally evolved. And that's going to dramatically change with the advent of AI design, not just for the capsid as we're doing it today at Dino, but broadly for all the different components of the therapies, enabling us to really have the maximal impact on patients.
I hope what the future holds are a better macroeconomic environment, lower interest rates, no wars in Ukraine. That part we can't control, but I do think in gene therapy what it'll show is what will emerge is numerous programs in the clinic with engineered capsids as the primary delivery vehicle and not wild type, and even products potentially approved and on the market to treat patients, and hopefully a resurgent view from investors about the potential for gene therapy and a renewed sense of interest.
Yeah, I think it's the same, better vectors, better payloads. I think the one thing in the next 5 years, surely we should aspire for is better regulatory clarity, because I think that will impact the approvability of number of programs ultimately for the patient. So I do think that that's a clarity that will emerge as more and more programs move through the clinic.
Maybe I'll start with a serious thought first and then end with a lighthearted one at the end, since there's only about a minute left. I do believe there'll be less companies in gene therapy. I think there's a bit of a flushing out that is happening right now, unfortunately, but I think it's healthy. It happens every few years. I think all of us have been in the space enough time. I do believe gene therapy and AAV will have a resurgence. I think if you listen to the types of numbers we are quoting, no matter whose number it is, this is not incremental change. It is many-fold, like orders of magnitude changes, right, improvement. So I think the majority of INDs will be new capsids, as an example. I believe there'll be at least 5 new approvals in AAV over the next 5 years. I think there'll be multiple takeouts, including more than $1 billion in takeout. On a macro note, which I like how you talked about that, I am pretty sure in 5 years, folks, the higher for longer term will be history. And I think the generalists will be back and all of us will be much, much happier. So.
Great note.
Hopefully.
So, all right, any closing thoughts? Otherwise, we're just about at the end of our session. So I'd like to thank all of you again for your participation and the fantastic discussion.
Thank you.